In this module, we will also talk about the range of fermentation product, as we talk about in a previous module about the third range of fermentation process, which is enzyme production. But in this, we will talk about biotransformation product. So, the fermentation process can be utilized for various type of reactions, in which we can use it for the conversion of molecules from one to another within an organism or outside the organism, either using by the cell as a catalyst or their product. As I told in a previous module that the enzymes can be produced through fermentation, but if we utilize those enzymes for the conversion of cellulose into glucose or starch into maltose, we can call as the bioconversions. So, there are number of chemical reactions in which the fermentation act as, you see, the help to convert those reactions, help in taking place those reactions. As we know that in chemistry, there are different processes: hydration, dehydration, hydrolysis, amination. Such kind of the reactions can be takes place during the fermentation process when mass culturing of the organisms happened. So we not focus upon the biomass production, not focus upon any metabolite production, but we are directly concerned with that what organisms can do during this fermentation process. In biotransformation processes, we add some substrate which is converted into other products through the organisms during the fermentation process. So you can see here the different examples, as I already told you, that starch into maltose, starch into glucose, benzaldehyde into alphenyl acetyl carbinol. In this slide, you can see that this is a very important compound which is called as L-ephedrine. L-ephedrine is also produced through fermentation process, which we called as the biotransformation of benzaldehyde to alphenyl acetyl carbinol. This biotransformation is carried out through the yeast fermentation. In this, when the yeast culture grow, when there is a suitable biomass of yeast during the fermentation process is achieved, and we know that yeast contain one enzyme which called as pyruvate decarboxylase. So, enzyme as a cell, either the enzyme of that PDC can convert the benzaldehyde into alphenyl acetyl carbinol. This alphenyl acetyl carbinol is the precursor of an important compound which we called as L-ephedrine. Before the fermentation process, this L-ephedrine is mostly extracted from an gymnosperm plant which we called as ephedra. There are a number of species of ephedra, but ephedra gerardiana is mostly used for the extraction of L-ephedrine. But with the discovery of this process that we can use yeast for the biotransformation of benzaldehyde to alphenyl acetyl carbinol, from the day to onward, this biotransformation method is mostly used for the production of L-ephedrine. So, along with this biotransformation, as I already told you, that for most of the microbial fermentation, glucose dissimilation occurs through the glycolytic pathway. A simple organic compound, mostly commonly generated, is pyruvate, or a compound derived enzymatically from the pyruvate, such as acetaldehyde. So, the acetaldehyde then can be reduced to NADH to ethanol, which is excreted by the cell. So, there is number of biotransformation examples. We use different microorganisms for the sake of bioremediation process. We can degrade some toxic compound into non-toxic compound by using this fermentation process. In various industries, effluent treatment is mostly use this biotransformation method by using the fermentation process.